Hello, I'm Katherine Naasko, a PhD candidate in soil sciences at WSU Pullman. I study the relationships between abiotic and biotic soil health indicators in deep soil profiles of dryland wheat in the Palouse River Basin and irrigated perennial tall wheatgrass in the Columbia Basin. Currently, I'm working on my dissertation research alongside Pacific Northwest National Lab researchers studying how abiotic soil health indicators such as soil moisture, soil pH, soil carbon and nitrogen influence soil microbial drivers of carbon cycling under limited moisture in deep soil profiles. Today, I will be sharing exciting novel correlations and important relationships between fatty acid biomarkers and pools of carbon and nitrogen, illustrated by the results of soil health indicator assessments in dryland winter wheat cropping systems in the Inland Pacific Northwest. This unpublished data is from the DSP for Soil Health project in cooperation with the NRCS. First, let me give a brief overview of the diversity in climate, landscape, soils, and cropping systems in the Inland Pacific Northwest. In the image shown here, you can see a depiction of the distribution of 10 soil orders that exist within Washington state. That's right, 10 soil orders out of 12 that exist worldwide can be found in Washington. The diversity in soil types is reflective of the heterogeneity that exists within the inherent abiotic soil formation factors of climate, landscape, and parent material. There is a precipitation and climate gradient across nearly the entire state, with variance in landforms including the ocean, mountains, desert, fertile rolling hills, and so on. The area outlined in gray here was previously and recently studied through the Regional Approaches to Climate Change, or REACH, in the Inland Pacific Northwest. This area is home to 6 soil orders and encompasses a wide range of dryland winter wheat production systems. Beneath the surface, there are silt loam soils internationally recognized for their fertility, and this is all connected by the Palouse River watershed, which you can see here. Lastly, the area follows a regional agroecological classification scheme in which there are 3 primary dryland cropping zones. Pictured here, you can see the yearly rotation schedule followed on a regional basis. Moving from west to east with greater precipitation and increased capacity of the soil to provide essential ecosystem services, cropping sequences intensify and therefore diversity increases. Just to make sure that all of these maps kind of make sense in y'all's heads, here I have outlined the Snake River in light blue on all 4 of the maps. So in this area, there is heterogeneous diversity into the soil profile and across the landscape with rolling hills and variable management systems. These pictures here are from the soil health in-field assessments and sampling time that we had in late 2018. It was a super fun, memorable, and like life lesson learning experience. So with this diversity that is within the area, there is also diversity into the soil. So management-based degradation is reflected in a loss of soil structure, reduced soil organic carbon concentrations, soil acidification, loss of topsoil, heavy erosion losses. So many producers have shifted towards more sustainable management practices, such as reducing tillage and incorporating residues to mitigate the soil losses towards the soil surface and build soil organic carbon in the surface soils. While many of these practices are implemented for their ability to protect the soil surface and lessen land degradation on the landscape scale. The abiotic and biotic drivers, long-term and short-term effects of these practices extend deep below the surface in dryland cropping systems. Deep-rooted crops such as winter wheat rely on deep reserves of carbon and mobile nutrients throughout their entire rooting zone several feet deep into the soil profile to fully take advantage of the minimal yearly precipitation. This allows for multiple season-long uptake of water, mobile nutrients, deeply exudated rhizole deposits, and complex associations with more mineral-dominant horizons deeper in the soil profile. In these areas, deeper soil health metrics allow a more comprehensive assessment of the productivity in the system with respect to abiotic and biotic drivers that are present in the variable intensities as you move through the soil profile. With all this being said, there is a lack of studies examining soil health indicators with depth across variable management systems in the Palouse region, as previous literature has focused on assessing relationships between abiotic and biotic indicators of the surface samples. So some studies use like the top 10 or the top 20 centimeters, but other using as little as the top 5 centimeters. So this is a trend, you know, the more than just in the Pacific Northwest, as historically soil and crop sciences have focused on the soil surface, as they are thought to be kind of reflective of the lower soil profile and are easier to sample. And let's just be honest, it's way easier to analyze data when you have just one soil horizon. However, in recent years, there have been studies showing that sampling with depth into the soil profile and also representatively sampling from the field based on your experimental questions or grower concerns. Recent studies have shown that those are really important to consider when you sample. So today I will present data from a soil series-wide assessment of dynamic soil properties with depth in the Palouse soil series. So there were three main objectives to the sub-study I will present today. First, the partnership with the NRCS laid out the experimental variables of soil depth, land use, and precipitation within a soil series. So that kind of helped formulate the first objective, to compare these indicators of deep soil cores across variable land use and the precipitation gradient within a single soil series to see how much variability or similarity there is within the samples. So secondly, after becoming familiar with the data set, after lots of time in R, I wanted to illustrate the importance of sampling deep within the soil profile and the use of segmented soil sampling techniques based on the study design and which research questions are being asked. And lastly, I would like to show some interesting and novel correlations between pools of soil organic carbon, including smaller, more labile and more microbially kind of relevant pools of PoxC and water extractable organic matter, and how those compare with phospholipid fatty acid biomarkers. So, our study took place at 7 sites at 5 farms near Pullman, Washington and Moscow, Idaho. This map here shows kind of the same regional depiction of Washington State and the REACH study area. And then in red here, you can see the Palouse soil series. So the blue boxes kind of represent the same approximate area within the state. And then let's see, so more specifically, we sampled at 2 long-term local producer fields, 2 Washington State University managed farms, and a long-term strip trial with variable tillage intensity at the University of Idaho. These can be broken down further into 2 low precipitation sites in red, one under no-till management and one under tillage, and then 3 moderate precipitation sites in green, and those have either no-till management, till management, and then there's also 1 perennial grass site that was planted 20 years ago, which serves as like a control or like a baseline kind of comparison, and then we also have 2 strips, 1 no-till and 1 till, which receive the greatest precipitation, and those are managed by the University of Idaho, and those are in blue. So then the blue are University of Idaho, like I just said, green are managed by WSU, and then red are the producer sites. So we also have kind of a difference in like field scale. The red are actual fields, the green are kind of more long-term experimental sites, smaller fields, and then the strip trials in blue. So there's multifaceted differences in this study that we have to account for when we think about the data and how to interpret it. So in total, we sampled 105 soil cores, which were homogenized into 21 soil profiles, which were then further split into 7 depth increments and homogenized by soil horizon, which I will describe in further detail right now. So from each of the 7 sites, we had 3 pedons for a total of 21 pedons. Within each pedon, I split our soil cores into 6 depth increments, as you can see here, by taxonomic horizon designation. So the 30, 60, and 85 centimeter depths varied within each core as the topography of the Palouse results in variable depths in these lower horizons, but they were, pretty close to 30, 60, 85 centimeters as the breakdown. So these soil samples were analyzed for a wide array of abiotic soil health indicators as well as a few microbial indicators. So more specifically, we looked at infiltration in the field using 2 methods, a Cornell and single ring infiltration. We looked at wet aggregate stability using the slow wetting and fast wetting techniques. We analyzed our samples for macro and micronutrients, soil pH, soil moisture. So we also analyzed our samples for a number of pools of soil organic carbon, soil organic matter, and nitrogen, including these here. So soil organic matter, total organic carbon, and total nitrogen using combustion. Permanganate oxidizable carbon, where you basically oxidize the soil sample and calculate how much carbon has reacted with the potassium permanganate solution that you add. And then we have autoclave citrate extractable protein protocol. So PoxC and ACE protein are recognized as kind of more labile pools, more kind of associated with the microbial community, whether they were, produced by the microbial community, or whether they're more used by the microbial community, or whether those pools are higher. Does that mean that the microbial community is not using those as much? There's multiple different ways that they can be taken. So we measured all these different carbon pools that you can see here, including the water extractable organic carbon and nitrogen pools. So we had the cold water and hot water sequential extractions. And then in terms of the microbial data, we had beta-glucosidase enzyme activity that they did at Kellogg Soil Survey Lab, and then we also had phospholipid fatty acid analysis done by the Soil Health Assessment Center in Missouri. So, as previously mentioned, today I will be focusing on kind of the outlined areas here, so the carbon and nitrogen indicators, PLFA, and then the kind of upper soil depths, well, not upper, I guess upper and upper to 85 centimeters deep in the soil profile. So let's dive in. First, let me explain to you what I have tried to summarize in this figure. Here I have broken down the results by soil horizon, and you can see the average value of each indicator we measured with AP1A representing the 0 to 5 centimeter horizon, and then the AB representing the 60 to 85 centimeter horizon. So this is like if you're looking at the soil profile from the soil surface to 85 centimeters down, what were the average values of the indicators that we tested for. So in summary, average across all 7 sites, soil health indicators were significantly different by soil horizon, including indices of soil organic matter, total organic carbon, PoxC, hot water and cold water carbon, total nitrogen, ACE protein, hot water and cold water nitrogen, and fatty acid biomarkers, including specific biomarkers of gram-negative and gram-positive bacteria, anaerobic bacteria, eukaryotes, fungi, and actinomycetes. So we have quite a diversity in our data set, as you can see here. Soil pH significantly increased with increasing depth, whereas all of the other indicators significantly decreased, moving from 0 to 85 centimeters. You can see that the pools of carbon and nitrogen changed differentially with depth, signifying that soil characteristics do not actually respond the same to increased depth, and the dampening effect is indicator specific. So on this slide, you can see a summary of the analysis of variance in each indicator by factor of soil depth, mean annual precipitation, and land use, and the various combinations of those. So you can see in order to summarize the PLFA data for this table, I have calculated a couple of different ratios in addition to total PLFA, and so ratios of gram-positive to gram-negative bacteria, and also the ratio of fungi to bacteria. So let's see. As seen, total organic carbon and permanganate oxidizable carbon, cold water carbon, total nitrogen, ACE protein, cold water nitrogen, and PLFA biomarkers were significantly different by the mean annual precipitation gradient. Hot water carbon, cold water carbon, ACE protein, hot water nitrogen, cold water nitrogen, and PLFA biomarkers were significantly different by land use. Notably, all indicators were significantly different by soil horizon and by the combination of land use and mean annual precipitation. So that is like the second column and then the second to last column. You can see that pretty much all those were like P was, the significance was less than 0.01. So from the way our study was designed, when indicators are significantly different by the cross between land use and mean annual precipitation, a kind of reasonable interpretation of this is that the indicator is site-specific. So the indicators were site-specific and depth-specific, I guess you could say. But when site and depth were crossed, some of the significance went away, as you can see in the last column of this table. Now that you all have an idea for how the soil health indicators vary by depth, land use, and precipitation, let us look at how the indicators are correlated to one another. So here on this correlation graph, I have various PLFA microbial biomarker groups on the vertical axis and the carbon and nitrogen pools on the horizontal axis. As you can see, there are strong correlations across the board. The trend of carbon, nitrogen, and PLFA shows that surface horizons have generally greater values than deeper horizons, which coincides with previous published literature that explains that, carbon and microbial activity often lowers as you move into the soil profile with less inputs from above ground and below ground plant biomass and exudates, and as well as the above ground climate influences. So while the correlation between PLFA biomarkers and total organic carbon and total nitrogen are the strongest, there are more inferences and questions to be asked when comparing PLFA to more unique carbon and nitrogen pools, such as the more microbially relevant pools of PoxC and ACE protein, as well as the water extractable organic pools. So, previous studies have illustrated how soil, bacterial, and fungal communities are responsive to shifts in the carbon to nitrogen ratio as bacteria are less adapted to more extreme environments as compared to fungi. More specifically, bacteria require more nitrogen per unit biomass carbon and are less resistant to dry, warm, acidic soils when compared to fungal communities, which can survive in more stressful environments. So while you'll know that, correlation doesn't mean causation, this project and these results have opened up a world of questions and hypotheses for me to work on here at PNNL while I'm getting some multi-omic, metagenomic, and metatranscriptomic data about the soil microbial community. So let's, for now, just take a closer look at how these correlations kind of differentiate by land use and mean annual precipitation. So here on this slide, you can see the correlations between PoxC and various biomarkers of the soil microbial community, including gram-negative bacteria in red, gram-positive bacteria in yellow, anaerobic bacteria in green, and actinomycetes in blue, as well as fungi in purple. So you can see that the correlations actually become stronger for gram-negative and gram-positive bacteria as you decrease the level of disturbance, moving from conventional tilled wheat to perennial grass. A similar relationship is seen when you move from low precipitation to high precipitation. The correlation between PLFA microbial community biomarkers and PoxC increase in correlation value. So these relationships show that different microbial members are correlated to different pools of soil organic carbon. Now, it's not really possible to discern whether the microbial groups are present at higher levels because there are higher concentrations of these carbon pools, or if the carbon pools are higher because there's more microbial community members who are contributing to the production of that, or maybe they're not using it quite as much and that's what's causing higher levels in the soil. So it's really difficult to do any further interpretations other than seeing which biomarkers are correlated to which carbon pools under certain conditions. So on this slide, you can see the correlation between the carbon and nitrogen ratio and the previously discussed microbial groups. The correlation is actually the strongest for actinomycetes, a little bit weaker correlation for anaerobic bacteria, and then a little bit less for gram-positive and gram-negative, and the least correlated is fungi. So the soil to CDN ratio has been previously correlated to the ratio of fungal to bacterial biomarkers, as well as gram-positive and gram-negative ratios. But unfortunately, this data set, that did not happen within this data set. So here on this side, you can see the correlations between the ratio of gram-positive to gram-negative bacterial PLFA biomarkers and the water-soluble carbon to nitrogen ratio, which I calculated using the sum of cold water carbon and hot water carbon divided by the sum of cold water nitrogen and hot water nitrogen to formulate the ratio of water-soluble carbon to nitrogen. So under conventional till, there was a pretty strong correlation between the ratio of gram-positive to gram-negative bacteria and water-soluble carbon to nitrogen. This correlation was not present when examining the no-till wheat sites. This correlation came back slightly in the perennial grass sites. So this is just kind of a look at, even though kind of on a bigger scale, it doesn't really look like there is variability within the data set. If you look at certain ratios and look at the relationship between certain microbial biomarkers and certain carbon and nitrogen pools, there are differences. So one of the additional kind of primary questions of this research project was, well, does sampling depth matter? So here you can see the PCA, principal component analysis diagram of the soil health indicators that I included in the study that I've talked about so far. So this top left picture shows the distribution of the results of the combined PLFA data and soil health indicators. It looks a little ugly, just to be completely honest, and it's kind of hard to figure out what's going on. So let's pop onto what happens in the 5 to 10 centimeter horizon. So as you can see, the way that you interpret these principal component analysis plots is the more closely points are to one another, the more closely they are related. So, and these results I have grouped in the PCA plots by site. So each of the 7 sites has its own circle. And you can see that there are some more defined relationships and overlap between the sites when you compare the 5 to 10 centimeter horizon that were not present when comparing the 0 to 5 centimeter horizon. This trend continues and more defined relationships appear when you get down to the 10 to 30 centimeter horizon. And even more so, things become defined from the 30 to 60 centimeter horizon. If you look at the explained variance between the PC1 and the PC2 on these plots, you can see that as the depth of the samples included in the PCA plot increase, so as the depths increase, the explained variance going on the first principal component axis, it actually decreases. So more of the variance is explained in kind of some of the other indicators that weren't really quite as determinate in the top 5 centimeters of the soil profile. So, and then lastly, we have the 60 to 85 centimeter plot. This one excludes eukaryotes, fungi, and anaerobic bacteria, those were not present. So this one we expect to see kind of a dramatic shift because it's not inclusive of, you know, all of the data points that were in the other plots. So kind of another way to like do a broad scale analysis of how the data varies by site is to use kind of a summing technique to sum how the averaged indicators across each horizon, as you add them as you go down to the soil profile, how the soil health indicators change. So from the 0 to 5 centimeter horizon, you can see here on the top left again, same as the last graph because it's the same data and the same depth increment. So and then 0 to 10, once again, things become a little bit more clear and the relationships become a little bit more distinct and we have more variability that is explained in 2 axes versus just having, you know, 99% of the explained variance in the first axis on just the 0 to 5 depth increment. So then when you include 0 to 30, it becomes kind of even more defined. The trend continues, 0 to 60 and 0 to 85. So basically, the more depth that you include, kind of the more defined these relationships become into the soil profile. And when you compare the principal component analysis diagram of including, you know, 0 to 85 centimeters down in the soil profile with the one that is 0 to 5 centimeters down in the soil profile, there are, you know, hardly any similarities in the distribution of the patterns and also the distribution of the indicators that were included in the PCA. So in summary, soil depth was a consistently stronger driver of abiotic and biotic indicators of soil health compared to land use or mean annual precipitation. Sampling technique and depth matter. There's variability between soil horizons individually, and the greater the depth that you include, the more defined kind of grouping of sites become, which points to the kind of conclusion that a lot of these indicators are site-specific and can be compared on, you know, a very specific basis. So also, lastly, soil microbial fatty acid biomarkers are in fact predictable using pools of soil organic carbon and total nitrogen. As seen, you know, PLFA microbial biomass is most strongly correlated to total organic carbon and nitrogen pools rather than pools of PoxC and the water extractable. However, they're all kind of co-correlated with one another. So if you have to really kind of weigh the pros and cons of each protocol and decide which one you want to move forward with and which is best for your study, what you're looking for. So certain microbes are, you know, more strongly correlated to certain pools, as we saw in the graphs, and some of these correlations kind of weaken as the level of disturbance increases, whereas others get stronger. So I hope you enjoyed listening to my presentation, and there should be papers out pretty soon, but I hope you learned a lot. Thank you. Bye.